Okay, so neurons and action potentials. To start, we're going to go over what a neuron actually is. Neuron is essentially the basic structural unit of the nervous system, and it is a nerve cell. Okay, so now we know what it is. Let's actually draw it. So we have our center part, which is known as the cell body outside, and we have each of these little dendrites sticking out. Now, obviously, this is not a very super accurate drawing. However, it gives you a rough idea. And connected to the cell body is the axon. Yeah. And that is connected to the axon terminal, which is this portion here. Now, this axon terminal is the region of the neuron that connects to the dendrites of other neurons and allows for communication between neurons. And that connection is called a synapse. So a synapse essentially refers to when an axon terminal of one neuron and a dendrite of another neuron will connect with one another. Now, to reiterate that one more time, essentially, dendrites are regions that receive information, and axons are regions that transmit information across the neuron. So now you have a rough idea of what a neuron looks like. Obviously, there's more sections and individual parts within the neuron, but we're going to go into detail on that a little bit later. But for now, this gives you a good rough idea of what a neuron looks like. Okay, so now we're going to talk about action potentials. Now, an action potential essentially allows for the transmission of signals between neurons. Now, this transmission of signals results in the electrical membrane potential, which is the difference in charge between the inside and outside of the neuron, to rapidly change. Something that helps me remember is that the action potential is the process and the membrane potential is the variable that is changing. Okay, so to actually break down the steps of the action potential, I'm going to first explain it to you and then I'm going to draw it. So the first stage is when the neuron is at its resting state. So we'll show you resting state here. Now, this resting state could be, between -50 to -90 millivolts. However, just for the sake of an example, we'll say it's around at -65 millivolts. Now, the next stage involves some sort of stimulus. It could be a sensory stimulus, or it could be a excitatory postsynaptic potential, which causes the membrane potential to increase to what is known as the threshold potential. Stimulus -65 millivolts, so the membrane potential to increase to around -55. Now, once the membrane potential reaches this threshold potential, the voltage-gated ion sodium channels will open. Now, what this means is that it will allow for a large influx of sodium to enter into the neuron, which will cause rapid depolarization of the membrane potential. And what that means is that the membrane potential will become increasing very, very positive very quickly. So I'll just write that down. Will become very positive. Now, at around +30 millivolts, when the membrane potential increases to around +30 to 40 millivolts, the voltage-gated sodium channels will actually close, and the potassium-gated ion channels will open, and this causes sodium to stop entering the cell. However, potassium will begin to leave the cell, and this causes what's known as repolarization. Depolarization essentially means when the cell membrane potential will become increasingly positive, and polarization is when the cell membrane potential will become increasingly negative. To actually explain this even more, from around -55 millivolts to +30 millivolts, this depolarization will occur. And then after that, potassium channels will open, and this is at about, again, like +30 to 40 millivolts. And at this point, membrane potential will become very negative. It'll actually become so negative that it'll actually decrease. It'll actually go lower than that of its original resting state and that lowest point that it reaches after the potassium channels open is a stage known as hyperpolarization. And then finally it will return to its resting state. Now, hopefully that was explained well enough that you can somewhat understand that. That may seem like a lot of information, so what I'll do actually now is I'm going to draw that for you, and that'll kind of just really solidify that knowledge, because it is a lot of content. Okay, so to do that, I'm going to draw a millivolt scale. Okay, cool. Okay, cool. So this portion here is minus 65 millivolts, which, like I said before, is the resting state, which is right here. So at this resting state, now some sort of stimulus will occur. So that could be a sensory stimulus or a excited postsynaptic potential. But whatever stimulus it is, it causes an increase in membrane potential. Now, when it reaches a certain, we're going to call this threshold potential here. And yeah, we'll say this is about, say, minus 55 millivolts. By the way, this is not going to be to scale. However, it'll give you a rough idea of what it looks like. So it reaches this threshold potential, and this is when, like we talked about before, these voltage-gated sodium channels will open at around minus 55 millivolts at that threshold potential. And this is when a rapid depolarization of the cell will occur. So rapid influx of sodium will cause this depolarization. And the membrane potential will go to, this could be around, we'll say plus 30 to 40, because it varies. So it could be between that range, and that's at this portion here. So right here is, this whole up arrow is depolarization. And as it reaches this point where, and now as it reaches plus 30 to 40 millivolts, the sodium channels will close, and simultaneously these potassium channels will open, which is this repolarization stage, and the membrane potential will become very polarized, so it will become very negative. So it will go down like this. So it will decrease like this to a level that is below that of the resting state. And we'll say this portion, this is repolarization. This section here is the hyperpolarization stage. And now to reiterate that, the membrane potential is more negative than that of its original resting state, and that's hyperpolarization. So you can see that this region is actually more negative than this original resting state. And then the membrane potential will return to that of its normal resting state. So we'll say this is about minus 65 millivolts. Now, this is a rough drawing of what an action potential actually looks like, but in summary, essentially, it starts off at its resting state, increases to its threshold potential due to some sort of stimulus. The sodium channels will then open, causing rapid depolarization. Eventually, at around 30 to 40 millivolts, the sodium channels will close, and the potassium channels will open, causing potassium to leave the cell, which makes the cell more polarized, more negative, which causes the electrical potential to decrease, and it'll decrease to a point at which it's below its original resting state, which is known as hyperpolarization, and then back to its resting state. Okay, that's the action potential process, and you know what a neuron looks like. So now we're going to talk about how does that actually happen within a neuron. So the first key point is that as the depolarization phase occurs, the repolarization phase will follow it almost, and it'll block the spread going backwards. I think almost a better way of thinking about it is, imagine that this was the axon, the depolarization, okay, the repolarization is happening here, and the depolarization is happening in this portion here. Hopefully that makes a bit more sense. So if you had to visualize this rectangle as an axon, The depolarization is the green portion that's happening in front, and the blue portion is the repolarization stage, which is happening just behind that depolarization, and it is preventing the spread backwards. Okay, we're going to keep talking about action potentials for a little bit, and actually talk about myelinated axons. So let me write that down just to explain that. So it's referred to as myelinated axons. Now, what myelin essentially is, it's around 80% lipid, which is fat, and 20% protein. It essentially insulates axons and allows for action potentials to propagate. So myelin increases the speed of nerve conduction, and it's present around nerves in the CNS where it insulates axons. Okay, now to show you how it actually helps, it's in a process known as saltatory conduction. So imagine this rectangle here is the axon, and each of these little rectangles I'm drawing each of these little rectangles I'm drawing within this axon is what is called the nodes of Ranvier, which essentially are regions that the action potential will jump between. So you can see here, it connects like that. And this rectangle I've drawn is the axon. Alright, so this process of saltatory conduction is where the action potential jumps from one node of Ranvier to another, and it goes along the myelinated axon. Rather than traveling smoothly down the whole axon, it jumps across these myelinated sections, which dramatically increases conduction speed, and signals will move 100 times faster in myelinated axons compared to unmyelinated ones. And this is the process of saltatory conduction. So this is how an action potential moves along an axon. Now, the final portion of this video, I'm going to talk about synapses. Now, I briefly mentioned synapses in the earlier section of the video, and a synapse is essentially a connection between the terminal of one axon, which, let's see if I still have that drawing, but yes, I do. So it's a connection between a terminal of one of the axons, which is this region here, these little things here, and the dendrites of another axon. That is a synapse, and that's how communication is transmitted between axons. What is actually transmitted in the process of a synapse? So that is a neurotransmitter. So a neurotransmitter is synthesized in the cell body of the neuron. It's transported to the end of the axon and stored at the end of the presynaptic terminal, which is the axon terminal, in small packages called vesicles. Now, when an action potential reaches the end of the axon, so when it moves all the way along the axon, the calcium will move in, which allows for exocytosis, where the neurotransmitters are released in small packages known as vesicles. Now, these neurotransmitters will diffuse across the synaptic cleft and bind with receptors on the postsynaptic membrane. To even just break it down again, like the calcium is released into the cell, triggers exocytosis of the neurotransmitters, which are released in packages called vesicles, and these vesicles will diffuse across the synaptic cleft to the receptors on the dendrites of the other neuron, and they will be broken down by enzymes, or they will undergo reuptake and cause another action potential to occur, and it could cause another stimulus in that way. Two most important neurotransmitters, and they're called glutamate, is one of the important ones, and it essentially opens Na+ channels, causes EPSPs. EPSPs are essentially excitatory postsynaptic potentials. And then GABA opens chlorine channels. Then this causes IPSPs. And maybe you can guess what that is, but that is a inhibitory postsynaptic potential. Now, a balance of these determines whether an action potential will be inhibited or whether it actually goes through. And finally, very final concept is for coding information. and I just want to briefly mention two ideas, which is temporal summation and spatial summation. So temporal summation is when multiple signals from the same presynaptic neuron occur in rapid succession, and spatial summation is when signals from multiple presynaptic neurons arrive at different locations on the postsynaptic neuron. So what does that even mean? Let's actually explain that a little bit more. So yeah, like I said here, let's say this is your dendrite and your cell and multiple signals. I'm going to draw these as red. But let's say you have multiple signals from the same neuron. This is temporal summation. And then you have summation, which is when signals from multiple presynaptic neurons arrive at different locations. So that could be here, all over the place anyway. But it's from a multiple different presynaptic neurons, and that is called spatial summation. Now, you might ask, why is temporal and spatial summation important? It's like, why is the difference important? So, when in temporal summation, when one synapse sends multiple signals in rapid succession, the postsynaptic potentials add up over time. And this determines ultimately whether a neuron actually fires or not, because temporal and spatial summation allow the neuron to integrate all that information to decide whether it should start an action potential or not. Yeah, essentially temporal summation is important because it responds to repeated inputs over time, and it's useful for signals that are fast, rhythmic, or ongoing. Spatial summation responds to multiple sources firing at once, and is useful when signals come from various neurons or locations. And yeah, they're both methods to fine-tune control, adapt to different contexts, and create speed and precision in creating action potentials. Okay, that was the first video on neurons and action potentials. Hopefully that, made a little bit of sense and helped you understand these concepts a little bit more. Definitely helped me understand it even more as I taught it. So yeah, that's all for this video.